Assalamualaikum warahmatullahi wabarakatuh. I am Makaampang Nisrin Iki and I'm going to give you some knowledge about vesicle transport. Let's start with the cell. So the cytoplasm of eukaryotic cells contains of a membranous organelles including endoplasmic reticulum, Golgi complex, and lysosome that are functionally and structurally related to one another and to the plasma membrane. So these various membranous organelles are part of a dynamic, integrated endomembrane network in which materials are shuttled back and forth as part of transport vesicles that form by budding from one compartment and fusing with one another. Transport vesicles move through the cytoplasm in a directed manner, often pulled by motor proteins that operate on track formed by microtubules and microfilaments of the cytoskeleton. The biosynthetic pathway of eukaryotic cell consists of a series of distinct membrane-bound organelles that function in the synthesis, modification, and delivery of soluble and membrane proteins to their appropriate destinations in the cell. So, as illustrated in the photo, materials are carried between compartments by vesicle or other types of membrane-bound carriers that bud from donor membranes and fuse with acceptor membranes. A biosynthetic secretory pathway moves proteins from their site of synthesis in the endoplasmic reticulum through the Golgi complex to their final destination, an organelle, the plasma membrane, or the extracellular space. Whereas an endocytic pathway moves material in the opposite direction from the plasma membrane or extracellular space to the cell interior. So cargo is directed to its proper destination by specific targeting signals that are part of the proteins themselves. So the endoplasmic reticulum or the ER is a system of tubules, cisternae, and vesicle that divides the cytoplasm into a luminal space within the endoplasmic reticulum membranes and a cytosolic space outside the membranes. So the endoplasmic reticulum is divided into two broad types, the rough ER or the RER, which occurs typically as flattened cisternae and whose membranes have attached ribosomes, and the smooth ER or the SER, which occurs typically as tabular compartments and whose membranes lack ribosome. The function of the RER includes the synthesis of secreted proteins, lysosomal proteins, and integral membrane proteins, while the function of the SER vary from cell to cell and include the synthesis of steroid hormones, detoxification of a wide variety of organic compounds, and sequestration of calcium ions. This picture is a schematic diagram showing the stacks of flattened cisternae that makes up the rough ER. The cytosolic surface of the ER membrane contains bound ribosomes, which gives the cisternae their rough appearance. Most of the lipids of a cell membranes are also synthesized in the endoplasmic reticulum and moved from the site to various destinations. As each protein is synthesized in the rough ER, it becomes inserted into the lipid bilayer in a predictable orientation determined by its amino acid sequence. This orientation is maintained throughout its travel in the endomembrane system as illustrated in this picture. The carbohydrate chains, which are first added in the endoplasmic reticulum, provide a convenient way to assess membrane-sidedness because they are always present on the cisternal side of the cytoplasmic membranes, which becomes the exoplasmic side of the plasma membrane following the fusion of vesicle with the plasma membrane. The Golgi complex functions as a processing plant, modifying the membrane components and cargo synthesized in the endoplasmic reticulum before it moves on to its target destination. The Golgi complex is also the site of synthesis of the complex polysaccharides that makes up the matrix of the plant cell wall. Each Golgi complex consists of a stack of flattened plate-like cisternae with dilated rims and associated vesicles and tubules. Materials enter the stack at the seize phase are modified as they move by vesicle transport toward the opposite or trans phase. As they transverse the stack, sugars are added to the oligosaccharide chains by glycosyltransferases located in particular Golgi cisternae. Once the protein reach the trans-Golgi network or the TGN at the end of the stack, they are ready to be sorted and targeted for delivery to their ultimate cellular or the extracellular destination. Most or if not all of the vesicle that transport material through the endomembrane system are encased initially in a protein coat. Several distinct types of coated vesicle have been identified. So the three best studied coated vesicle are the following. So COP2 coated vesicle carry materials from the endoplasmic reticulum to the Golgi complex. COP1 coated vesicles carry materials in the opposite retrograde direction from the Golgi back to the endoplasmic reticulum. Clathrin-coated vesicle carry materials from the TGN to the endosomes, lysosomes, and the vacuole, plant vacuole. Proposed movement of materials by vesicular transport between membranous compartment of the biosynthetic secretory pathway. The three different types of coated vesicle indicated in this schematic drawing are thought to have distinct transport routes. COP2 coated vesicle mediate transport from the endoplasmic reticulum to the endoplasmic reticulum Golgi intermediate compartments and Golgi complex. COP1 coated vesicle return to proteins from the endoplasmic reticulum Golgi intermediate compartments and Golgi complex to the ER. COP1 coated vesicle also transport Golgi enzymes between cisternae in a retrograde direction. Clathrin coated vesicle mediate transport from the TGN to endosomes and lysosome. Transport of materials along the endocytic pathway is not shown in this drawing. So, second drawing. So, schematic drawing of the assembly of a COP2 coated vesicle at the endoplasmic reticulum. Assembly begins when SAR1 is recruited to the ER membrane and activated by exchange of its bound GDP with a bound GTP. Cargo proteins of the ER lumen or the red spheres and diamonds bind to the luminal ends of the transmembrane cargo receptor. These receptors are then concentrated within the coat vesicle through interaction of their cytosolic tails with components of the COP2 coat. Endoplasmic reticulum resident proteins are generally excluded from the coated vesicle. So those that do happen to become included in a coated vesicle are returned to the ER. So each compartment along the biosynthetic or endocytic pathway has characteristic protein compositions. So resident proteins tend to be retained in the particular compartments and retrieved if they escape to other compartments. The sorting of the proteins in the biosynthetic pathway occurs to a large extent in the last of the Golgi compartments, the TGN. The TGN is the source of vesicle that contain the particular membrane protein that target the vesicle to a particular destination, and you can read the next. Vesicle that bud from a donor compartment possess specific proteins in their membrane that recognize protein located in the target or acceptor compartment. So the docking of the two membranes is mediated by different proteins and regulated by a large family of J proteins called RABs. Fusion of donor and acceptor membranes is mediated by V snares and T snares, which interact to form four-stranded bundles. So, according to this model, RAB proteins on the vesicle and target membrane are involved in recruiting tethering proteins that mediate initial contact between the two membranes. Two types of tethering proteins are dipeptide, highly elongated fibrous protein, and multi-protein complexes. So, during the docking stage, leading up to membrane fusion, a V snare in the vesicle membrane interacts with the T snares in the target membrane to form a four-stranded helical bundle that brings the two membranes into intimate contact. In the cases described in the text, SNAP 25, one of the T snares is a peripheral membrane protein that is bound to the lipid bilayer by a lipid anchor rather than a transmembrane domain. So SNAP 25 contributes 2 helices to the 4 helix snare bundle. So these are proposed in step the targeting of transport vesicle to target membranes. Lysosomes are diverse appearing membrane-bound organelles that contain an array of acid hydrolyses capable of digesting virtually every type of biological macromolecules. Among their numerous roles, lysosomes degrade materials such as bacteria and debris that are brought into the cell by phagocytosis. They degrade aging cytoplasmic organelles by a process called autophagy, and they digest various macromolecules that are delivered by a way of endosome by receptor-mediated endocytosis. In vertebrates, lysosomes play a key role in immunologic defense. Lysosomes also play a key role in the organelle turnover, that is the regulated destruction of cells' own organelles and their replacement. So, during this process, which is called autophagy, an organelle such as the mitochondrion shown in this photo is surrounded by a double membrane structure, or what we call the phagophore, to produce a double membrane sequestering vesicle called an autophagosome. Once formed, the outer membrane of the autophagosome fuses with the lysosome, generating a structure called an autolysosome, so in which both the inner membrane of the autophagosome and the enclosed contents are degraded. The product of these degradative reactions are made available to the cell. The vacuoles of plants carry out a diverse array of activities. So vacuole serves as a temporary storehouse for solutes and macromolecules. So they contain toxic compounds used in defense. They provide a container for cellular wastes. They maintain a hypertonic compartment that exerts turgor pressure against the cell wall. And also they are sites of intracellular digestion mediated by acid hydrolysis. So, endocytic pathway, so endocytosis and the phagocytosis. So, endocytosis can be divided broadly into two categories: the bulk phase endocytosis and receptor-mediated endocytosis. Bulk phase endocytosis, or also known as pinocytosis, is the non-specific uptake of cellular fluids. So, receptor-mediated endocytosis, or the RME, in contrast, brings about uptakes of specific extracellular macromolecules following their binding to the receptors on the external surface of the plasma membranes. Receptor-mediated endocytosis. So, this sequence of a macrographs shows the steps in the uptake of yolk lipoproteins by the hand oocyte. So in A, the proteins to be taken into the cell are concentrated on the extracellular surface of an indented region of the plasma membrane, forming a coated pit. So the cytosolic surface of the plasma membrane of the coated pit is covered with a layer of bristly, electron-dense material containing the protein clathrin. So, and B, the coated pit has sunk inward to form a coated bud, so as you can see in the picture. So, and C, the plasma membrane is about to pinch off as a vesicle containing the yolk protein on its luminal. So, previously extracellular surface and clathrin on its cytosolic surface. So, in picture D, a coated vesicle that is no longer attached to the plasma membranes. So the next step in the process is the release of the clathrin coat.